Okay, it is 3 o'clock, and so we will go ahead and get started. Today we have Seth Tolley giving his PhD proposal presentation that's required by the department, and he is looking at ear photometry and maize breeding. And with that, Seth, I'll turn it over to you.
All right, thanks, Richard. So yeah, again, my name is Seth Tolley. I'm a second-year PhD student with Dr. Teinstra. I got my undergraduate degree at the University of Illinois, graduated from there in 2017. And then I came over to Purdue for my master's degree, which I did with Dr. Mohammadi, where I did some root phenotyping and other traits similar. And now I'm working on generally, the general theme of my thesis for my PhD is the use of the ear photometry platform in maize breeding. So with that, we'll get underway. So just to give a brief outline for where we're going in the next hour, I'd like to give a little bit of a background on maize hybrid breeding and especially look at the heterotic group formation for the US and CIMMYT breeding programs. We'll transition into looking a little bit more into ear photometry and give a little bit of background on the platform that we use and also other platforms that have been proposed. And then we're going to dig into some of my research chapters, spending the most amount of time on the first chapter, which looks at the heterotic patterns of temperate and tropical maize using ear photometry. And then I have 3 other chapters that we're going to look at, which uses preliminary data and then concepts that we're proposing to use for future work. And we'll just finish up with some conclusions. So just to start us off, this is the slide that I think everybody in corn breeding always starts off with. But as you can see here in this on the left, we have year on the x-axis and yield in bushels per acre on the y-axis. You can see that in maize breeding, we've gone through 3 distinct stages. The first one being open pollinated varieties, which was characterized by being relatively low yielding. Additionally, you can see in this picture then on the right that it was also characterized by being very heterogeneous in the look of the field, where there was a lot of variability, both in terms of plant height and also in terms of relative maturity. The next phase was the double cross era, which is described here in this image on the right. You can see that you start off with 4 inbred lines, you cross 2 of these together in a pair, and then the other 2 together in a pair, forming 2 single cross hybrids, and then you cross the single cross hybrids together to form your double cross. And then the final era, which we've been in for about 80 years now, is the single cross era, which has shown an increase of yields of about 1.9 bushels per acre per year. And that's illustrated in this picture on the right, where you can see an inbred line on the left crossed with an inbred line on the right. And then the hybrid of these 2 inbred lines is shown in the middle. And it has far greater yield capabilities than either of these 2 inbred lines. And that phenomenon is known as heterosis and hybrid vigor. But one thing that we know is that when we cross inbred lines together, not all are going to show this great degree of yield increase as we see in this image. So that led the US maize breeding to identify and classify these heterotic groups. And you can see that this is generally done in this picture, this cluster analysis done by Michael in 2008 from the University of Illinois, where he shows the separation between the stiff stalks and the non-stiff stalks. Some recent publications, such as this one here, which you can see from Dr. Rochford's group in 2017, also looks at the ident classification of a heterotic group, as well as the non-stiff stalks and the stiff stalks. And this is largely done because the idents show they're often used, and also because they show similar performance with the non-stiff stalks and the stiff stalk in terms of yield. Then moving away from the US heterotic groups into CIMMYT heterotic groups, the CIMMYT hybrid maize breeding program began in 1985. They started out evaluating many combinations of heterotic groups, but when you're breeding for combining ability between heterotic groups, it gets really difficult when you have multiple groups. So currently they have simplified it a bit to where they have two main heterotic groups, the Tuxpanio, which they classify as heterotic group A, which is a dent variety kind. And then you have the non-Tuxpanio, which is classified as B, and it's a flint variety type. And one thing that we're going to dig into a little bit more in the first chapter that we're going to talk about is that these are often difficult to classify when you use genomic information. And that's been seen in previous literature and then also something that we're going to talk about. So then moving into discussing ear photometry. So this platform was patented by Pioneer, now Corteva, in 2009. And there are 4 general steps that we followed. First of all, we had unprocessed images that were imaged just from one angle. These are classified and segmented using supervised classification that separates the important features from the background of these images. These are processed using a proprietary software from Corteva, and then the data is output as a text file, and then it's compiled together. To show this more generally and visually, you can see that here's an unprocessed image. We have a mask that we used to separate the kernels and the cobs from the background. Then we further separated the kernels from the cobs. And then finally, what we ended up with was a processed image where we were able to extract about 25 unique ear characteristics. But while Pioneer's patent was one of the first to recommend the use of this technology, more recently there have been other universities and public organizations. So I believe we have the University of Illinois, Wisconsin, and then CIMMYT where these 3 publications come from. On the left, you can see different kinds of platforms that they used. And then on the right here, you can see the correlation between the reference measurements to the prediction of ear photometry traits. And what you can see here in all 3 of these, in ear length, for kernel weight, and then for kernel count, these platforms have previously shown that ear photometry is a viable option to accurately assess yield components. So digging into this first chapter then, which we actually just recently have submitted for publication, we had 3 primary objectives. The first was to also validate the use of ear photometry. And what made this a little bit unique is that we wanted to not only do it on a temperate set of germplasm, but also to do it on a tropical set of germplasm as well. We wanted to characterize the relationship of these traits and the heritability of all of our ear photometry traits. And then we finally wanted to describe heterotic patterns that we see in temperate and tropical maize using ear photometry. So this experiment was performed in 2017 and 2018 in a randomized complete block with 2 reps. We looked at the test cross performance of 286 temperate inbred lines, which were both founding public lines and then also XPVPs. And then we also looked at 247 drought tolerant maize for Africa, which we call DTMA inbred lines from CIMMYT, and test cross performance to PHPO2, which is an iodate line. We also looked at the test cross performance of 297 temperate inbred lines in test cross performance to 2-face, which is a stiff suck line. In 2017, we sampled 10 years per plot, and in 2018, we sampled 5 years per plot, and in total, we looked at more than 21,000 ears. To do validation, we took reference measurements for kernel weight, kernels per ear, yield on a per ear basis, and then finally yield on a per plot basis. So here on the right, you can see our ear photometry platform that was donated to Purdue by Pioneer Corteva in, I believe, 2016. For this study, we had a couple of different data analysis that we did. The first one was we predicted our BLEPs using the following equation, where we had the phenotype was equal to the mean population plus the random effect of the hybrid plus the fixed effect of the year plus the random interaction between hybrid and year. And then we had the fixed effect of rep nested within year. Finally, we just had the error term. We determined broad sense heritability using an entry mean basis and a plot mean basis, where the difference between these is the use of the number of years and the number of reps in the denominator. We further did principal component analysis, both on the ear photometry traits to characterize their relationship, and then also on the 533 inbred lines used in this study using the genomic information that was available. Finally, to declare significant difference between these heterotic groups, we used analysis of variance and Tukey's test. So to start off with then, we had 533 inbred lines that were evaluated in this study. Here in this graph, you can see principal component 1, which explained 20.2% of the variation in the genomic information on the x-axis, and then principal component 2, which explains 16.6% of the variation in the genomic information on the y-axis. Along principal component 1, you can largely see the separation between the tropical germplasm, the DTMA germplasm from CIMMYT, from the temperate US germplasm. Then along principal component 2, you can see the separation between the stiff stalks and the non-stiff stalks and iodines. Principal component 3, which I don't show here, is what separates the non-stiff stalks from the iodine groups. One interesting thing, which I've kind of alluded to already, was that in doing this analysis in the principal components that we evaluated, we never really saw the difference between the heterotic groups of the CIMMYT germplasm, even when we looked at them separate from the temperate germplasm. So rather than looking at these 3 heterotic groups, AB and AB, by themselves, we just grouped them all together and called them DTMA. So the first objective that I talked about was we wanted to validate the use of our ear photometry platform. So in these 4 graphs, you can see our reference measurements on the x-axis, and then you can see the photometry estimates on the y-axis. And what we found was that kernels per ear and yield were both accurately assessed when done by ear photometry. We did notice a general trend, though, that kernel weight was a little bit lower. Finally, then in looking at panel D, you can see our reference yield on a plot basis as measured by a combine. And then we had adjusted phenotype, or adjusted yield on the y-axis. And this was adjusted for stand count. We wanted to look to see if there was a difference between the temperate and the tropical germplasm. And what we found was for kernel weight and yield, we did have a lower prediction accuracy in our tropical germplasm compared to our temperate germplasm. Then we wanted to look at the heritabilities of these ear photometry traits. So we looked also at our reference measurements that we made, but for this talk, I'm going to specifically just keep an eye on the ear photometry traits. What we found was that yield, as estimated by the photometry platform, had one of the lowest heritabilities on an entry mean and plot mean basis compared to our other traits that we evaluated. But when we moved to traits such as ear length and ear perimeter, as well as the traits more related to average individual kernels, we found a much higher heritability compared, especially with yield, but then some of the other traits as well. This is used to look at the relationship between our traits. So in panel A on the left, you can see a principal component analysis. And then in panel B on the right, you can see a clustering analysis where we identified 5 main groups using the Ball Hall index. The 2 largest clusters identified were those that were related to individual kernel attributes, and then those that were related to more broadly ear characteristics. And you can see that yield was nested within this bottom cluster. Using principal and component analysis, you can kind of look at this data in a little bit of a different way from clustering. So we have principal component 1 here, principal component 2, and principal component 3. Our interpretations were checked to make sure that they were orthogonal and that they were valid in that way. Along principal component 1, you can see that it was generally separated based on their correlation to yield, which is how this principal component analysis was clustered. Principal component 2 was a contrast between traits that increased kernels per ear and traits that increased kernel size. And then principal component analysis 3, principal component 3, we found it was a contrast between traits that increased the overall size of the ear versus traits that increased the amount of the ear that was filled with kernels. So the final objective of this work was then to look at the heterotic patterns that we were seeing with our testers. And so while we do have these large tables that we can come back to if there are questions on this later, I'd more like to use these images that we created because it shows it visually in a little bit nicer of a way. So you can see here that, before I dig into it, let me just say that these ears are mean ears for each of these heterotic groups. They aren't cherry picked just to show the results that we were looking for. So what we found was that the non-stiff stalks had the significantly greatest yield, where the stiff stalks was significantly worst. The ident was found to be an intermediate between both of these heterotic groups. An interesting note was that kernels per ear and ear length were also found to show this similar trend to yield, where the non-stiff stalks was the greatest, the stiff stalks were the lowest, and then the idents were in the middle. However, our kernel specific attributes such as mean kernel size, kernel weight was not found to be significantly different between any of these heterotic groups. So that kind of told us that perhaps these traits increase kernels per ear and ear length, including yield, were affected more by heterosis than the kernel attributes. So then looking at the heterotic patterns with PHPO2, the ident tester, we found a similar story where the non-stiff stalks and the stiff stalks were not significantly different in terms of their yields. However, when you then looked at the ident lines, that's when you saw a significant reduction in yield. This was again based on kernels per ear and overall ear length rather than the individual kernel characteristics, as was previously mentioned when we looked at it in combination with two-face, the stiff stalk tester. When you add in the DTMA material, you can see that it was not significantly different from the non-stiff stalk or the stiff stalk heterotic groups. There was, however, an interesting trend that there was a significant decrease in kernels per ear, which was more associated to the fact that we had a larger proportion of the tip kernel abortion than in the temperate heterotic groups. Let me go back. What was interesting, though, is that the DTMA material made up for this in terms of its yieldability, because the individual kernel characteristics were significantly greater than our temperate heterotic groups. So then that leaves the first chapter behind us then. From here on out, it's a little bit more of preliminary research and what we're kind of proposing to do for the rest of my thesis. So the second chapter for now is called Genomic Evaluation of the Temperate and Tropical Maize by Ear Photometry. And we had 4 main objectives for this work. The first one is to do genomic evaluation of the inbred lines and the ear photometry traits. We want to do GWAS, genome-wide association study, And then we want to do more genomic prediction, looking specifically at single trait analysis versus multi-trait analysis. Just to give a very brief background on some of the studies that we're going to do. The first one that we're employing is GWAS, which is a genome-wide association study. This uses historical recombination to find associations between markers and traits. How I typically think, what I typically think about when I think of GWAS is these Manhattan plots, as you can see here on the right. So on the x-axis here, you have chromosome number, and on the y-axis, you have the negative log p-value. And so each one of these dots represents a different marker that we've used in our study, with those markers falling above the negative log p-value of 4 being deemed significant, and then even those that are in green were significant based on FDR. The other thing that we wanted to use was genomic prediction. And so for genomic prediction, you train a model that uses your genetic information that then you can use to predict the phenotypes of individuals where you have genotype information, but you don't have phenotype information. So the materials and methods for this study, it's actually the same study that we already talked about in the first one. The only difference between this slide and what I've already presented is that we're also planning to do a validation study in 2020 where we've also extracted 10 ears per plots from this year. Further materials and methods for this. For our genotype information, we started out with about 750,000 single nucleotide polymorphisms, or SNPs, for the 533 inbred lines. We filtered these SNPs in P-Link, which is useful software. And we filtered for minor allele frequency of less than 0.05. And then we filtered out SNPs that were missing in more than 25% of genotypes. In total, we ended up with about 53,000 SNPs for all 533 inbred lines. These were imputed in Beagle, and we produced hybrid genotypes using Tassel. So just to dig into some of our preliminary results that we're showing, this on the left is an admixture model where we use a k-value of 4. So there are 4 ancestral populations in this image. And this is used to tell the background of your inbred lines. So in this, you can see the 533 inbred lines on the x-axis. Each of these is a different bar plot, and you can see the amount of the population that comes from a certain background. Then the other thing that we wanted to do for the genomic evaluation was we wanted to look at the genetic correlation of the 25 ear characteristics that are evaluated using ear photometry. And so far, I just show a little bit of preliminary work. I've started out by just doing the first 10 traits. And so a larger study then will use all 25 of our ear characteristics. We performed GWAS using GAPIT and R, specifically using the farm CPU model. And here are just some of the summary statistics that we analyzed from this work. We had a certain number of SNPs that were significant for each of these traits. We're just showing 6 of them here to represent the larger body of work. You can see that there was variability in the greatest single SNP, which was adjusted with a certain SNP in yield explaining almost 10% of the variation. And then we showed the total amount of variation. So for example, for tip kernel abortion, the trait on the bottom, you can see that we explained almost 30% of the variation in the phenotype using these 14 traits. And then finally, we just have listed some of the candidate genes that we found using Maize QTL database. So then moving into our single trait genomic prediction. So far, we've just done it again for these 6 traits, but we split our data set into 75% training and 25% validation. We repeated this 20 times, and you can see for each of these traits, we had a range of prediction accuracies or correlations to the real phenotypes that we had. Now, taking this a step further, which I'm not really going to show in a slide, we're going to do a multi-trait genomic prediction as well. So that'll take advantage of the variance covariance matrix between our phenotypes to implement it for genomic prediction, but that is yet to come. Third chapter that we're going to be talking about today is yield component prediction using integration of the environment, genomics, and phenomics. In this study, we have collected longitudinal traits from UAV platforms, and we're going to use these for yield prediction. And then in the future, we're going to try to integrate phenomics and environmental data to do crop modeling. This work was performed this year in 2020, and it'll also be done in 2021. We evaluated a subset of the hybrids that we did in the first study, where we had 10 temperate and 10 tropical inbreds that were evaluated for their test cross performance with PHPO2. And this was done with 3 reps. Throughout the growing season, we collected 19 UAV flights, which were done roughly on a weekly basis, and we collected RGB, multispectral, and LiDAR information. The plan will be to gather weather data from local weather station, and we have also collected 10 ears per plot to do ear photometry. Just to show what I've been working on with this so far, it's just been image processing. So on the left here, you can see a LiDAR image captured on June 17th that's been georeferenced and stitched together. You can see that same thing for RGB on June 17th. And then just to show how that's transformed throughout the growing season, then you can look at what we see from July 8th for the LiDAR and the RGB. So the goal will be to extract useful information from these images that we can use to help in our prediction of yield. So finally, what we're going to talk about is the use of ear photometry for the development of heat-tolerant maize for Asia. So the objective of this work is to identify inbred lines that have enhanced heat tolerance that we could incorporate into breeding pools. And then number 2, we want to validate phenotyping strategies that can be further used. And thanks to COVID, we don't have data from this yet. So rather than talking about data that we have collected, I just wanted to provide a general background of this project and the reason for this project. And so just to start out with talking about South Asian maize production, it represents about 3.5% of the global maize production. What's unfortunate though is that yield is roughly half of the worldwide average. So when we look at consumer trends and how they're shifting over time, it's anticipated that demand for maize in South Asia is going to outpace production by 2025. So if you look at agriculture in this region, they have 3 main growing seasons. This first one from late June to September, where their major crop is rice. You can see that this growing season is characterized by moderately low temperatures and then adequate rainfall. Have another growing season from October to early March, where they grow small grains. This growing season, while also having fairly moderate temperatures, is characterized often by drought. And then you have this final growing season, which is extremely difficult, where you have both extreme temperatures and then also less water availability. And so this is one of the reasons why a heat tolerance project would be vital for this region. Just to look at the impact that heat stress can have on yields, this was a study that looked at 20,000 yield plots across Africa. And their key finding was that for each degree day spent above 30 degrees Celsius, you're reducing yields by about 1% when you have optimal rain-fed conditions, and then by 1.7% under drought conditions. So if we look instead at how much more extreme the temperatures are in this region, there's definitely a reason for concern. But we really think that ear photometry can be a platform that can help us to visualize some of these lines that show enhanced heat tolerance. Specifically, just looking at this picture, you can see that there's a wide variety of phenotypes that you can see. And I'm really interested to see how some of our traits help us to show these differences in these lines. So this experiment is done at the ICRISAT campus in Hyderabad, India. And it'll also be done at another site, which is about 40 miles to the north. In total, we have 80 hybrids that are being grown, 40 heat tolerant, and then 40 heat susceptible. This will be grown in a randomized complete design with multiple reps. And then ideally, it'll be repeated under each of their growing seasons, optimal, the managed drought, and then also under the heat stress. So just to provide some very general conclusions from this work, we really believe that ear photometry is a technology that can be used to efficiently measure yield components. And then also we believe that the implementation of ear photometry can be used in a variety of ways in maize breeding programs. So with that, I just want to thank Dr. Teinstra for taking me on, first of all, but then for all of his guidance with this project. My committee members, Dr. Brito, Dr. Crawford, Dr. Vine, and Dr. Wong. And then just really want to thank the Teinstra Lab and members of it, and then also friends of the department that have really helped me with this work as well. So with that, I'm happy to take any questions. And I just thank you for your time and for your attention.
If you have questions for Seth, unmute and say who you are and then ask your question.
Hello, this is Tony. Hi, Seth. How are you?
Hi, Dr. Vaughn. I'm doing well today. How are you?
I'm doing reasonable under the circumstances, I guess you would say. I am curious a little bit about the relationships of the ear photometry to the kernel weight parameter. At the beginning of your talk, you provided an example from published studies indicating pretty strong relationships, but then when you got into some of your own results, the relationship of ear photometry to kernel weight was fairly poor, I would say. And so I was wondering, in these stronger relationships, whether that was because it was primarily a hybrid study with a relatively high, I would say, kernel weights, whereas when you got into the inbred studies, you were maybe dealing with an overall lower range or more variability because it was an inbred, or because it was a composite of 582 inbreds.
Yeah, that's a really good question, Dr. Vine. Thanks for asking it. So the main difference between this platform and previous platforms that have been described, and these are both positives and negatives, like a lot of things are. The positives of this platform is that we're only imaging these ears from a single angle, and then we're never shelling the kernels in terms of photometry. A lot of these previous studies have imaged these ears from multiple angles, and then they've also, for looking at their estimates for kernel weight, they've shelled the ears, and then they've looked at the shelled kernels rather than actually using the kernels that were on the ear. And so to me, that's the biggest reason why our correlations are so much lower than what's been previously reported. Like I said, though, it is a trade-off because what's previously been reported might not be as feasible for a large-scale breeding company. You know, I don't know how feasible it would be to image ears from multiple angles and then also to shell the ears. just to take the image of the kernels that are shelled. But it's absolutely a trade-off between what you're willing to sacrifice in terms of correlation.
As with any technology, there are calibration opportunities. And so I'm just sort of wondering, is there something that could be tweaked within the calibration to provide a more robust kernel weight estimation?
That's a really good question, and not something that I've thought about a lot. One thing, if I can pull up this next slide, when we look at the difference between our, now we're flying way past it. When we look at the difference between our temperate and tropical material, I would say one of the possibilities that we have to improve our platform a little bit, especially in comparison to what I've shown here, is to have a better prediction of our tropical material. And I've thought about reasons why the tropical material could be less accurate than temperate material. Another thing that I want to mention in this study, this was we used the mean of the 10 ears that were harvested from the plots for our validation. Whereas in the future, I would definitely recommend looking at each ear individually. And I think that'll also show a much higher correlation than what we show in this manuscript.
Could you give me some idea as to what variation there is in the girth of the cob itself and how it could perhaps, if you had, just a fixed estimate of kernel weight based on an assumption on the cob girth, that might be something that's changing across genotypes, that relationship. So in other words, we all know that there are hybrids that have relatively deep kernels, and then there are hybrids that perhaps have a wider girth of a cob and a fairly small depth of kernel. And so I'm just kind of curious, what do we know about the cob itself if all that we're doing is looking at, the exterior of an ear?
Yeah, that's a really good question too. I can't really give you a range of values for the cobs. That's not really something that I can recall.
Okay.
But it is something that would be interesting to look at for further studies, absolutely.
And then, as you know, probably where I'm going with this is because of the class you took with me, but tell me a little bit more about how you selected the 5 or the 10 plants when you took those on to a yield estimate. And was that yield estimate per plant, or was that yield estimate per unit area?
Yeah, absolutely. So we did.
You probably knew that this was coming.
Yeah, had to ask, I'm sure. Yeah. So in 2017, we took 10 ears, and in 2018, we took 5 ears.
Right.
These were representative for the plots, which, talking about your class, I know that you would prefer that they would be consecutive ears rather than representative ears. So for further research for this study that we did in 2020, we did do consecutive ears, but for this first study, we did representative ears that were not necessarily consecutive.
All right. And then just a final question is basically, in an ear photometry study, what do you do with barren or nearly barren ears, and how does that affect how you do the evaluations, I suppose? Like surely with some of these genotypes, especially if you get into heat stress, you're going to have barren ears. So what's your modus operandi in that situation?
Yeah, so far we've looked at hybrids from Indiana, so we haven't had ears that have been barren, such as what you saw in the
That,
in this picture right here, like these are much different from what we have been doing ear photometry on currently. So it will definitely provide an interesting challenge. So what you heard me talk mainly about was yield, kernels per ear, and kernel weight. However, there were many other traits, such as tip kernel abortion, scatter, and some other traits that are more focused on the stress of the specific ears. So I'm really interested to see how those traits are going to be able to be implemented and used for more work, specifically on that heat tolerant project.
very good. Thank you, Seth.
Yep, thanks, Dr. Ryan.
Hey, Seth, this is Dr. Tynstrom.
Dr. Tynstrom.
I was wondering, we've been talking as recently as yesterday about layering research technologies on top of the experiment that you did this last summer. I was wondering from the standpoint, I was actually thinking of this the whole time you were presenting, and it's a follow-on of exactly what Dr. Vine just indicated. We're running these kernels through the VMEC, right?
Yes, that's correct.
Is it possible to take some of the kernel measurements from the VMEC and add them to or incorporate them into the prediction equation for seed weight to see how you might be able to improve some of those estimates as part of the 2020 experiment?
Yeah, I hadn't thought about using that information in that way. But I think that could be something that we could implement, which would be a good idea. So the VMEC, for people who don't know, you toss all the kernels on top and then they slide down, and as they slide down, they're imaged. So you get estimates of width, volume, many other traits related to these kernel attributes. It would be interesting to see if we would use some of that information if our accuracy would go up. And then we could, like Dr. Vine has already mentioned, then we could fine tune the platform from Ear Photometry even more. So it would be a good idea.
And you could check to see how that impacts your yield predictions, because at the moment, most of the yield prediction scatter seems to be driven by poorly estimated seed weights.
Definitely.
Good idea, Dr. Vine. Thank you.
Always a pleasure.
Other questions for Seth?
Can people outside the committee ask questions too?
Of course.
Yeah.
Okay. So Seth, can you go to the last slide when you showed the conclusions?
Yeah.
So in here, the first conclusion you are mentioning, is it the conclusion that you are intending to get after these PhD theses, after all experiments? Or is it what you already observed with the first project that you've done? So is it a wish that you are putting there, or is it set on a stone?
No, I would say that that's something that we've seen. So this first study, we have 25 ear characteristics that are measured relatively rapidly. I think about working with Blake or Rupesh doing their yield estimates in wheat, and it would take 5 of us months to do it, whereas for some of these, when we use ear photometry to make some of these estimates for maize, it's much faster than that for us. So I think it's definitely something that we've already seen.
Okay, so if you've seen it, then one thing that can, at least in this presentation and even in your manuscript, maybe you can allude to how much time does it save if you do this way compared to a situation where you want to do a hand measurement? Do you have an estimate of that?
It would be something to think about. The problem that I could see in trying to implement that is some of these traits, I don't even know how I would estimate it. If I think about scatter, which was looking at the uniformity of the ears, I have no idea how long that would take if I was to do that.
Okay.
but it is a good idea and something that could
Just for
really
example,
illustrate the importance
yeah,
of this
just
technology.
for example, if I were to do it by hand, this much time or this many manpower it would take, but by doing the machine, this is the benefit. The next question that I have is how likely it is that you can retain or use the data from India ICRISAT? Is it going to be a firm chapter of your thesis, or it is still you're thinking about it?
I mean, I would call it a firm chapter of my thesis.
Okay.
We aren't sure when we're going to be able to travel to India, of course, because of COVID. But for now, that's definitely one of the things that we have involved in.
So the reason I ask this question is that I was waiting to see how you define heat stress. Like, what is heat stress? You didn't mention anything in this presentation. And what is heat stress tolerance? How do we define these things?
Okay, yeah, I can talk about that briefly. So heat stress in Maize can be defined a variety of ways. In this study, we're specifically looking at heat stress right around the critical period. So we're reaching the maximum temperatures at ICRISAT right around the time of flowering. So that's how we're defining heat stress. Then heat stress tolerance, then for us in this study, would be those inbred lines that would be able to have a better yield performance in this high temperature environment as compared to some of the lower end.
So what you defined as heat stress is a heat event, right? What kind of biological effect does this event bring to the plant in the maize context, in ICRISAT environment? That's what I was trying to ask.
I think I understand your question. So stress in maize at a variety of different stages can have vastly different impact. So for example, let me just say that yield in maize, the yield components are determined relatively sequentially. From the overall ear size would be determined, and then you would determine the kernels per ear, kernel weight. So depending on when the stress was actually hitting the plants, you would be seeing different phenotypes just because of when the stress was hitting. So in this study, we're having the stress hit the plants right about at flowering. So we should be seeing, this should be hitting it right when we would be seeing some phenotypes related to barrenness of the ears and so on.
Okay.
Seth, do you remember the traits that Dr. Zaidi is outlined in that book that I sent to you guys as a PDF? I don't know if you've read that book. Mm-hmm. Can you describe the trait? There's maybe 10 different traits. Can you describe any of those as it relates to the stress at the critical period? What kind of things does he describe in that book as? What are the traits that are recorded in these trials?
Sure. Yeah. So they looked at plant height and ear height were two of the ones that they looked at. They then also looked into the amount of lodging in the plant, specifically in root lodging and stem lodging. I can't recall specifically any of the other traits that were specifically evaluated.
Some of the other characteristics that are described in this standard phenotyping protocol is tassel blasting. So do the tassels become blasted and sterile during development under these high temperature stress conditions? Leaf firing. So this would be leaves that are scorched during that stage of, and they're normally described as leaves at the top of the canopy. Senescence, so the leaves at the lower end of the canopy senesce prematurely. As well as these yield components that Seth has already described, and as well as anthesis silking interval.
Right. Yep. Thank you.
I have probably one final question, Seth.
Okay.
Is that okay? So in your GWAS table, when you identified or you drilled down to the candidate genes, you had cases from 1 candidate gene to about 14, 15 candidate genes. Can you describe or explain how you got from those GWAS hits into these and why you have 1 candidate gene for one of the hits and then few or a dozen for the other ones?
Yeah, that's a good question. So this was determined in Maize QTL database. You can see that some of the variation is just due to the number of SNPs that we evaluated for the study. So for plant yield, we only had 1 SNP. So of course, we're going to have less candidate genes than for tip kernel abortion, where we had 14 SNPs that were significant in the study. One thing that is shown in this specific table, I used an LD value, or I looked at a window of 100 KB surrounding these markers, where we've already talked a little bit, and that's going to end up being changed, where we're going to use maybe a smaller window to better reflect linkage disequilibrium in Maize.
So the reason I ask this is that because when Travis was doing his GWAS analysis, I had a discussion with him about that window of the genomic regions that you need to stay, go to left, go to right, and find that. So this is a kind of a LD matter, like how much LD persists around those regions. Have you done any LD analysis so far on this germplasm?
I have, yeah. From what I recall from our preliminary results that we have is we had an LD of about, maybe it was 12,000 kilobases, I believe, was an LD of about 0.2. And from what I recall, it was, I looked into Travis's paper to see how our results compared to what he's found, and I believe results were fairly similar.
Okay. Thank you, Seth. Thank you very much. Good work.
Thanks, Dr. Mohammadi.
I think Alankar has one more question.
Yeah, just a quick question. So have you evaluated if there was an increase in heritability from using the traits driven by photometry in comparison to manually count them, or that was not even an option? And another question in the same line is, how are those traits correlated either phenotypically or genetically to yield?
Mm-hmm. Yeah, both are very good questions. So the first one, for this study, it was unfortunately not possible to really look at the differences in heritability between the photometry estimates and our reference measurements. And that's because our reference measurements were either taken in 2017 or 2018, but not both. So we already had a bias because we weren't including that year effect in our variance component analysis when we looked at heritability. And I'm sorry, could you repeat the second question? I'm sorry.
Oh, the second question is whether I just want to know if you have estimated the genetic correlation between these traits and yield, to see if by any means they correlate, if it's a phenotype selectable component that would provide any downstream advantage to yield.
Yeah, so I've just started doing that with a preliminary analysis. You can see this genetic correlation for yield is the first trait here, and then we have the first 9 other traits from this study. And this is just preliminary. It was run using BluffF90, and I ran out of power on my computer. So I'm learning now how I can toss this up to the cluster to be able to do all 25 of my traits at the same time. So this is very much a preliminary analysis and a preliminary result of this study. But it's definitely a goal to be able to look at the genetic correlation of these traits. And I really think that'll be something that'll help us then further to do the multi-trait genomic prediction. Thank you for your question.
Seth, one last question before we finish, I think. One of the when you showed the PC1 and PC2 and PC3 for the genetic diversity of the temperate germplasm versus the tropical germplasm, you showed in PC1 that it's sorted temperate versus tropical.
Yep, I'll pull it up here really quick. Yep.
And what this graph suggests is that there's more genetic diversity in the temperate germplasm than the tropical germplasm. I was wondering if this the appearance of genetic similarity of the tropical germplasm was a function of ascertainment bias and the SNP calling against the B73 genome.
It's a good question. I haven't thought about that before.
And if you would have some ideas about other kinds of SNP calling strategies that might reflect something that doesn't have that kind of a bias.
I honestly, I don't have a very good answer for this, but I guess that's the point of proposal presentation. So that way I can look at this over the next 2 years of my study, because I do think that that's a really good point and something that I haven't thought of to this point.
I can help you with that a little bit, at least to get to the answer. So tell us a little bit about how did you produce this marker data?
So the marker data, it was sourced from Dr. Rochford's lab. And then for the temperate germplasm, but for the tropical germplasm, it was sourced from CIMMYT, from an online repository. We had, I believe it was build 2.7 for the temperate germplasm, and then for the tropical germplasm, I'm not 100% sure.
So let's imagine that we don't have a marker data, okay? Can you walk us through how do you develop marker data? Like you have 533 inbreds, right?
Right.
How do you make the marker data? What strategy do you use for making new marker data?
So you would extract DNA from each of these lines. You would sequence it using Illumina or one of the such. That would spit out some raw reads. Some of these would be overlapping. And then you would toss these up to look at where they would fall on a reference genome.
What is the reference genome? What is the reference genome?
And as Dr. Teinstra already alluded to, that's B73, which is a temperate inbred line.
Are there other genomes that you can also use?
I'm sure that there are, but I haven't looked into that at all.
Okay.
Yeah.
All right. Do you have an idea that if you want to utilize a genotyping platform or call a company to do a genotyping for you on all of your materials, do you have an idea of what kind of cost are you looking at? So that you can get a clean set of marker data that is not, for example, if you're sharing some data from Dr. Rochford and some from Dr. Teinstra, and then you want to combine them to get a common set out of them. If you want to develop a new data set, what kind of cost are you looking at, and what kind of benefits does it have for you?
Sure. So cost, the cost of SNPs has recently gone down. It would, of course, vary based on how many SNPs we had, and then obviously on how many lines we had too. I don't know the exact cost of, in terms of a per line estimate right now.
That's okay. That's okay. Yeah. But does it help to get around to bypass the ascertainment bias, at least to some extent? That is about the sampling thing that we have.
Yeah, it probably could. Then we could use a difference reference genome, I suppose. Yeah, it might. Yeah.
Thank you.
Yeah, thank you.
Okay. Seth, have you had enough?
Yeah. Are there
All
more?
right. Yeah, so we're at 4 o'clock. So unless there's anything last minute, we'll go ahead and end with that. We will be back next week with another presenter. Look for information on that early next week in your emails. And that is all I have. Everybody have a nice weekend.